Come in. So welcome, my name is Jamie Trickus. I'm from the University of Sydney, a member of the CRE, and it is my pleasure to chair this session on Emerge in Drug-Resistant TB. We're running a little bit behind time, so I do ask our speakers who are not pre-recorded to try and stick to time, and we'll hopefully have time for a few questions. So our first speaker is, or speakers, I think, we have Philipp du Cros and Catherine Berry, who will talk about RCT MSF clinical trials. Thanks, Philipp.
Great. So welcome back, and hopefully we can entertain with our duet with Catherine and myself, and on behalf of the many, many people involved with this work. And it has taken 8 years to come up with that very bad title pun. So I'm sure most of you would have seen this intriguing announcement from WHO that kind of left you wanting more. You know, perhaps there's promising news from this trial that has stopped enrolling early. So we wanted to talk a little bit about this trial and what we can tell you that's public at the moment, and still probably leave you wanting more. This story goes back about 8 years. A meeting not dissimilar to this, maybe a few less people. The one difference was that we kind of locked the door on the room until we came up with some new ideas, which took 2 days on a weekend. And some of us had the thought at the end of this that we could really have some principles to guide more revolutionary, is that a daring word, on sort of trial design for MDR TB. I guess the challenge for this group is I'd say that all of the MDR TB trials currently ongoing for treatment are probably predicated on similar groups at thinking from 8 years ago, so we can probably relook at this space. And so MSF was one group that came out and has been the sponsor for a randomised controlled trial. It's an adaptive trial design, so a phase 2-3 trial in adults with pulmonary TB, looking at 3 novel regimens. I'm not going to go through all the details because we've been given only a short slot. Apparently, if you don't have all the data, you don't get the long slot. That's a lesson for you. But the population that we're going to look at is adults, so over the age of 15, regardless of HIV status, with confirmed MTB, confirmed rifampicin resistance, and with the new definitions of pre-XDR, they could be included, or it could be fluoroquinolone sensitive patients, so a broad population. The study was conducted in 3 countries at 7 sites, and you can see that Belarus, South Africa and Uzbekistan have slightly different manifestations or populations of the MDR epidemic. But all of those countries from their standard programs do slightly better in their standard treatment programs than sort of WHO reported outcomes from MDR TB. The investigational arms that we're looking at has a backbone of bedaquiline plus pretomanid plus linezolid, the so-called sort of BPAL that you've heard from the NICS TB regimen, one arm with moxifloxacin, one with clofazimine, and one with just 3 drugs, and given for 24 weeks. So the differences compared to BPAL is that it's just given for 24 weeks, so not this 6 to 9 months, and the linezolid was dosed at 600 milligrams from the start, and then that could be lowered to 300 milligrams for the last 2 months. The control arm was the locally accepted standard of care that was consistent with WHO guidelines. And so what we can say, that's not a single regimen, that's obviously a multitude of potential regimens, which is the challenge of doing MDR trials compared against a gold standard. And so what we can say is that the sites all had access to bedaquiline and dolaminid, and they could include shorter MDR TB regimens. So stage 1 is equivalent to the phase 2 trial, and this part of the trial was having a primary endpoint looking at safety and efficacy data to identify which arms to take forward. Having said that, all patients who were enrolled would get the full treatment in this stage, but it was only the 8-week data that was looked at, and that was looked at when there were 60 patients enrolled in each of the 4 arms. And stage 2, more equivalent to a traditional phase 3 trial, would take forward 1 or 2 investigational arms compared with the standard of care, and looking at the primary outcome of the percentage of patients with unfavourable outcomes at 72 weeks post-randomisation. The trial design is a non-inferiority design, and using a conservative alpha to take into account the sort of multiple analyses and allowing for non-assessable patients, so aiming for 201 patients per arm in the stage 2. So just going through that again, because it's slightly different from a normal design, just to hopefully pictorially represent. So all patients who start enrolment get the full treatment, and they're all followed up for 108 weeks to get the secondary endpoints. The primary stage 1 data was looked at just to select 1 or 2 regimens to go forward for the full trial. And for the stage 2 design, it ended up with 1 regimen going forward. That was a 1-to-1 design, and the primary outcome is a composite endpoint of unfavourable, as listed there, looked at 72 weeks, with allowing a transition stage for ethics and all the rest. And so then, in terms of the regimen that was taken forward, it was the arm containing moxifloxacin, so B-palm, I guess. And if we look at the standard of care, you can see some differences between the countries. But of note, drugs like imipenem were available in 2 of the sites for patients with fluoroquinolone resistance, and the shorter regimens were used in 2 sites, one including amikacin, the other using all-oral shorter regimen. And at this point, I'm going to hand over to Dr. Catherine Berry to bring us home.
Okay. So I think for anyone that's been involved in clinical trials in drug-resistant TB, this is very much a marathon, not a sprint. I think 2017 isn't really the beginning, 2012 was, and getting to first patient was an achievement in itself. And really, at the beginning of this year, we thought we would still be in a position where we would finish recruitment sometime this year, and then the plan, as we've said, was to follow patients up to at least 72 weeks. But what I'm about to explain has changed that slightly. So the DSMB was given a data lock. So just to give some background, the DSMB was provided with efficacy data on a 6-monthly basis and safety data on a 3-monthly basis per their charter. They were given a stopping rule that was incredibly conservative, that allowed for the multiple analyses. And that was just supposed to trigger an opening discussion about whether to continue the trial or not. So at the data lock of 31st of December 2020, the DSMB had received data from 242 participants with late exclusions removed from the efficacy analyses. And when I say like late exclusions, if they weren't proven to have MDR TB, they were excluded from the analysis. And their recommendation, it came back to us in a letter, and that's the information we have at the moment. The sponsor doesn't have a final analysis yet. Based on their review, the DSMB recommended termination of randomisation of the trial. The recommendation was primarily based on the guidance from the DSMB charter, as I've outlined. Specifically, it wasn't allowed to be just a culture conversion or some other indirect endpoint. It had to be something that impacted clinical practice. So a level of evidence was achieved favouring the experimental arm, that's we're calling B-palm for the purposes of this. And the difference in proportion of unfavourable outcomes was primarily driven by a higher rate of treatment discontinuations in the control arm. Specifically, the DSMB communicated the following information to us. The deaths were favoured in the standard of care at 5-0, and there were no treatment failures or recurrence cases in either arm. That says relapse, but recurrence is the current information we have. And so on the 17th of March of this year, we terminated enrolment into the trial, having enrolled 552 patients into the study. With around 200 that will have reached 108 weeks by that point. We have a number of sub-study collaborators. Specifically, we're undertaking a variety of PK/PD studies, patient-reported outcome study, which should really, certainly in oncology trials, and we think also in TB, it will be really useful to complement adverse event data to really understand how patients experience the TB treatments, and then an economic evaluation study. So later this year, there will be a review of MDR TB guidelines, and this is a number of the trials that are either in late stage or will have reported by then. I think of note, we have information Xenix will report this year, and possibly Simplicity TB. But there are a wide variety of studies that will continue to provide important information in making a shorter regimen standard of care for drug-resistant TB. And this is the plan for analysis. Of course, we've seen already this year shorter regimens in drug-sensitive tuberculosis and come through, and better treatments for meningitis, and a variety of regimens both with operational research and clinical trial data, and it'll be interesting to see how those different information streams will help instruct the guideline committee. So our conclusions, we, as much as anyone else, has been surprised that TB-CRE has been stopped for efficacy. We realise that that's not without some hesitancy to do that. It's controversial, but the DSMB were emphatic and unanimous in their recommendation, and would have been difficult for us to go against that. Completion of data checking is ongoing, and our priority following these initial brief communications will be to submit data to WHO, but also for review in a scientific journal for peer review. And I think it's our expectation and hope that by the end of this year, most patients with drug-resistant TB will be eligible for a shorter regimen. And that's going to make treatment better for them, hopefully, but also for the research community as well. So this is the type of number of collaborators, and I think there's a common theme throughout all of these talks today about this is what you need to make these possible. And I think most of all, in some of these places, both research and tuberculosis are quite stigmatised and difficult, and the bravery of the people living with TB can't be understated. So thank you, and happy to take questions if there's time.
Thank you, Catherine and Philip. Time for one or two questions.
Andrew Burke, Brisbane. Can I just ask about the decision to choose a moxifloxacin regimen to take forward, and why did that one stand out compared to clofazimine or?
Yes, I can't go into all the details on that, but quite a difficult decision. So I think what we can say is that for the pre-specified criteria, all three arms met those criteria. So we will be able to have data from the trial on all three arms and not just on the one arm that went forward. And particularly because of delays in shifting to stage 2, we had a lot more patients enrolled in all arms than we'd initially planned. So that's a positive made out of a negative. In terms of the kind of decisions that we were looking at is a little bit looking at the interim data that we had, a little bit looking at some data from qualitative research, again, a plug for qual researchers around which side effects are particularly problematic for some of the drugs. So in some countries, clofazimine is particularly reviled. And also taking into account the early data and what we could see at the time from other studies. So one of the members on the steering committee is TB Alliance, so they were also sharing their ongoing results as well.
Philip, I just wanted to ask about the adverse events, which clearly informed your choice to a large degree as well. But was there any discussion around rates of QT prolongation, like we've discussed, and whether levo should be the quinolone to consider instead of moxi to be combined with bedaquiline if you go with a single regimen?
Yeah, so when we were first designing the trial, we had a lot of discussion around different sort of regimens, and you'll notice that there's a different MSF trial that has chosen different regimens, and I think that's a good thing for the evidence base that we need. We favoured moxifloxacin because of thinking about maybe some data leaning towards it being more efficacious. But I guess the area that's complicated is if we talk about levofloxacin versus moxifloxacin, when we're not necessarily comparing equivalents of dose. So we're using moxifloxacin low dose, 400 milligrams. Levofloxacin, are you using it as standard dose, or do you mean 750? Do you mean 1,000? And what's the QT prolongation for levo if you bump the dose to sort of an equivalent efficacy dose of moxi? I think is still up for question because the studies haven't really compared sort of equivalent doses on that. But for good or bad, we chose moxi.
Last question.
Thank you. Just in terms of the methodology of your analysis and the decision to stop the trial, was the comparison including people from the control arm in the first phase as well as the people in the comparator arm, like the control arm in the second phase, or did you only compare people who started recruitment in the second phase? Because I just wonder whether like in some say, for example, a Bayesian adaptive trial, you might sort of use all of the evidence from all the phases in making those decisions.
Yeah. So just to state, so the first, the only analysis the sponsor saw was the phase 2 study, or the first stage, and all those patients were also included in the stage 2 part of the trial. So all of those ran through, and they were part of the data that was provided. So it was more, we had rules to stop any arms if there was a signal to suggest that there was a major problem with them, but we were taking them through otherwise. So all of that information was provided to the DSMB.
Okay, thank you very much.